Hello students. Today we will start with Medical Microbiology 1 in that unit bacterial pathogens. Bacterial pathogens with respect to their classification and biochemical characters, antigenic structure, viability characteristics, pathogenesis, symptoms, laboratory diagnosis, epidemiology, prophylaxis and treatment. Okay, so in this bacterial pathogens, we will be starting with enteric pathogens. Enteric pathogens, E. coli, classification and biochemical characters, antigenic structure and viability characteristics. Okay, now enteric pathogens are what? Why are they named enteric? Because it's a microbe that affects the intestine and can make a person sick. Fine. Enteric pathogens. Now we will be studying 5 different pathogens in this: E. coli, Shigella, Salmonella, Campylobacter, Vibrio. Okay. Now in bracket here in objectives I have given in bracket Enterobacteriaceae. This is a family. This is a large group of heterogeneous group of medically importance, and it is a gram-negative enteric bacilli that exhibits morphological and biochemical similarities, and so they are grouped together in a large and complex family which is Enterobacteriaceae. Their natural habitat is intestinal tract of man and animals, though few species may occur in other parts of the body or plants and in soil and water. Fine. And some species are commensals and some are pathogenic for humans. Now, what are their characters? They are aerobic and facultative anaerobic, gram-negative, non-spore forming enteric bacilli. They are often motile by Pittrich's flagella or they are non-motile. Their size ranges 2 to 3 into 0.4 to 0.6 micrometers with rounded ends and ferment glucose with production of acid or of acid and gas. Fine. Now let's move ahead. Now E. coli. As we have started with E. coli, they are the member of Enterobacteriaceae family. They are aerobic or facultatively anaerobic gram-negative bacteria. Okay. Enteric bacteria typically enter the body through the mouth. Typically, they are acquired through contaminated food and water or by contact with animals or their environments or by contact with the feces of an infected person. These are the microscopic images and this is the antigenic structure image for E. coli. We'll talk on this later. Next. Now, unlike other coliforms, E. coli is a parasite inhabiting in human and animal intestine. Now, why I am using this word coliforms? What are those? Now, the oldest method for classification of enteric bacilli was based on lactose fermentation, and members fermenting lactose are called as lactose fermenters or coliforms. Okay. Next. Now this gens consists of 5 species, among which E. coli is the most common and clinically important species. We have already talked about Enterobacteriaceae family. In that species, we have E. coli, the most common. Next, Cultural characters. It's an aerobe and facultative anaerobe and can grow on ordinary culture media. Next, optimum growth temperature is 37 degrees centigrade. Next, on nutrient agar, the morphology of the pathogen will be colonies are large, thick, colorless, moist, smooth, opaque or translucent disc. Okay. On McConkey's, the colonies are rose pink due to lactose fermentation. And on blood agar, some strains show beta hemolysis. Now we will not go in detail. Next, biochemical reactions. They break down most of the sugars, lactose, glucose, mannitol, maltose. I'm sorry, there's a spelling mistake over here in glucose, with production of acid and gas. Okay. Now, typical strains of E. coli, they do not ferment sucrose. Next, now this IMVIC test. What is IMVIC test? Tests are a group individual which test is used in microbiology to identify an organism in coliform group. Understood? Now we are studying what? E. coli. It's a coliform. Fine. IMVIC stands for I. indole test, M. methyl red test, V. Voges-Prosker test, C is for citrate test. Now here is a small I in between. Small I, it stands for in, in, as the citrate test requires coliform samples to be placed in citrate. That's why it is I, small I and C. Okay. Now E. coli. Indole production and methyl red reactions are positive, but VP and citrate utilization tests are negative. This is its biochemical reaction. Okay. Next, antigenic structure. Now, Kaufman in 1947, he published a diagnostic scheme for E. coli based on their presence of antigens O, H and K. O is somatic antigen, K surface antigen, H flagellar antigen and F fimbrial antigen. Got it? Now O antigen, somatic antigen. These are heat stable. These are heat stable. Next, they can be overcome inoculability caused by K antigens. Fine. Next, surface antigen, K antigen. The term K antigen refers to the acidic polysaccharide surface, capsular antigen, even if no detectable capsule is present. Now, K antigen is divided in two groups, group 1, group 2. Okay. Now, what is the role of K antigen? They cause O, antigen O, in agglutinability by preventing somatic agglutination of living strains by homologous antisera. And what's the second role? Act as virulence factor by impeding phagocytosis and protects bacilli from the killing action of antibody and complement. Understood? Now, intestine, it harbors several different serotypes of E. coli and most of them do not possess K antigens. Next, flagellar antigen, H antigen. These are thermolabile and more than 50 H antigens are described and all of them are monophasic. Okay. Next, fimbrial antigen, F antigen. Now, many Enterobacteriaceae, they contain fimbria. Okay, you can see the image here. See, got it? Now they have fimbria, the common pili which are chromosomally determined, present in large numbers and can cause mannose-sensitive heme agglutination, which are probably not related to pathogenesis. Fine. Now they show virulence factors. Type 1 fimbriae mediates addition of bacterium to human and animal cells that contain mannose residue. Fine. I'm sorry. Now, regarding viability characteristics, whatever characteristics of E. coli, their survival, habitat, everything, it comes under viability characteristics. Okay, fine. Still, if you have any kind of issue, you can ask me. Okay, thank you.